Hello. Welcome to Next in the series review clips designed to assist you in preparing for your Biology 3201 public examination. In this clip, we'll be taking a look at various theories that surround the origin of life, how life came to be on this planet. One of the first group of scientists to actually formulate hypotheses surrounding this were Haldane and Oparin. They believed that the early Earth consisted of what they termed to be a primordial soup. The early Earth consisted of organics, which existed in the aquatic environment, the seas, which formed a primordial soup. Inside of this soup, if you will, there were self-replicating chemicals that eventually coagulated, combined to form cellular life. They were the first to speculate on the spontaneous origin of life on this planet. The Haldane-Oparin Hypothesis proposes that the precursors of life's molecules formed from inorganic sources, and these inorganic sources underwent polymerization to form macromolecules. Such macromolecules included primitive versions of enzymes. Droplets, or as they term it, coacervates, formed and were enclosed by protein or lipid shells. These coacervates simulate life processes such as growth and reproduction, and by taking in selected materials they grow to a critical mass and then divide. Experimenting with their coacervates, Oparin constructed simple biochemical systems such as those containing replicating enzymes and nucleic acid elements. He managed to get the coacervates to duplicate some elementary biochemical mechanisms. So, some of the theories for the origin of life are as follows. Firstly, the Oparin-Haldane hypothesis, and they believe that life arose from chemical reactions that occurred in a highly reduced atmosphere, i.e., one that is rich in hydrogen-containing gases such as methane and ammonia. To provide credence to Oparin-Haldane's work, Miller and Urey conducted an experiment. They took gases and electricity and they made amino acids, which are, of course, the building blocks of proteins. So the geological evidence indicates that the Earth formed about 4.6 billion years ago. We know that the early atmosphere contained a number of gases: hydrogen cyanide, carbon dioxide, carbon monoxide, nitrogen, hydrogen sulfide, and water. Yet there was and is no oxygen or evidence of it in the early atmosphere. The Earth then cooled enough to form the first solid rocks, and about 3.8 billion years ago, the Earth cooled enough for water to condense, which were the formation of the first sedimentary rocks. So the Oparin-Haldane hypothesis that conditions on primitive Earth synthesized organic compounds from inorganic ones. Miller and Urey, they did experiments to determine if conditions could possibly produce organic compounds. They took a mixture of gases that were deemed to be present in the Earth's early atmosphere. They exposed electricity, which would simulate lightning storms. Cold water caused condense to forming droplets, and there were liquids containing amino acid and other organic compounds found in the residue. This showed that an amino acid, an organic compound, could be synthesized from inorganic ones through conditions, as a result of conditions similar to that which you would find on the early Earth. Another theory that assists in explaining how life arose on this planet is the PANSPERMIA theory. PANSPERMIA suggests that life's seeds came from outer space and planets exchanged life. In short, PANSPERMIA means seeds everywhere. PANSPERMIA suggests that life could have existed on other planets and moved to Earth. We know that 7.5% of rocks from Mars have reached the Earth. The rocks would travel between less than 100 years to 16,000 years and more to get to Earth. Some of the proponents of this idea, PANSPERMIA, include Salles-Guillaume de Montlevin, who proposed life came from the Moon. H.E. Richter suggested life came from meteorites and comets, and Svante Arrhenius, who came up with PANSPERMIA. There is evidence for it. We know that bacteria can indeed survive harsh environments as found in space. Ultraviolet radiation, the bombardment of proteins, and the cold, they can certainly survive that. We know that meteorites contain life. There's amino acids which are left-handed, which are different from those we typically find here. Bacteria, carbon, they could be protected inside rocks. We know that bacteria can live for a long time in a dormant state. Mars would be a safer environment than Earth. There's less bombardment, less gravity. So Mars could have been the site of the bacteria's original creation. It wasn't as hot as Earth in its early development. And it may have had oxygen back then when the Earth did not. Another theory is Gaia, as proposed by Jim Lovelock. Lovelock worked out that if he was on Mars wondering about life, he could just look through a telescope and see evidence that the gas in the Earth's atmosphere are in a persistent state of disequilibrium, or being driven by living organisms. Together with Lynn Margolis, they developed the idea of symbiosis, or the Gaia theory. In 1974, they stated the original hypothesis: the biosphere, atmosphere, oceans, climate, Earth's crust and biota, living organisms, is regulated as a homeostatic mechanism in conditions comfortable for the living organisms. Organisms working together. We cannot dispute the idea of intelligent design either. The intelligent design theory claims that the universe, or parts of it, have been designed by an intelligence, a divine being. It's possible to detect evidence of design by intelligence. We have extremely complex life systems organisms. We can use an explanatory filter. How is the design process followed? It's certainly complex with specified information. Intelligent design holds that with the amount of biological complexity on the planet, it cannot be generated solely by natural processes. The heterotroph hypothesis, another theory for life's origins, is that, and the strength of this theory hypothesis is that it gives the first forms of life a source of food that doesn't come from living things, the primordial soup. Firstly, the ability to self-organize. With already formed building blocks of soup and the source of energy served to help drive them in increasing complex forms, in the 1950s, experiments confirmed that this process could begin. Lightning, confined to a soup's bottle, which stimulate production of the complex materials. As a result, all the various forms of energy on the current Earth have been tested with varying results. The leading contender for life organizers are the hydrothermal vents, which you find at the bottom of oceans. There, water, through mixing with magma, releasing hot soup materials even today. There's an energy source, the heat. Once soup, now magma, and perhaps most importantly, it's a stable, long-lasting ecosystem and a place to work the bugs out of the earliest living systems. The ability to reproduce is explained by the heterotroph hypothesis as well. As the early molecules grew, only those who could make and spread themselves had any possibility of future. On today's Earth, the DNA code is used to store the information, but it does need to make proteins it actually runs on. The first systems probably weren't DNA or protein molecules. There are theories that try to express problems, but the leading current theory is that the first really complex systems were RNA-based, or the RNA world hypothesis. RNA has coding abilities and some protein-like activity, and it isn't difficult to see how a DNA-coded protein system could grow quickly from RNA ancestor. And the ability to evolve. If you have a planet-wide ocean full of self-organizing molecules that can reproduce themselves, you have a competitive system where selection could take place. This stage of molecular evolution would have favored those who could work most efficiently or best accumulate building blocks or reproduce the fast, or work with other molecules in a cooperative fashion, possibly linking DNA and RNA together. These unified systems might work even better with some confinement and protection. So if we look at symbiogenesis, symbiogenesis is a theory of evolution. It argues that acquisition and accumulation of random mutations or genetic drift are not sufficient to explain how new inherited variations occur in evolution. With reference to this theory, new cell organelles, new bodies, new organs, and new species arise from symbiosis. Here, independent organisms merge to form composites. This challenges some of Darwin's standard ideas. To some degree, Darwin emphasized competition as the primary driving force of evolution. Symbiogenesis emphasizes cooperation can also be important to the process of evolution. It was first formulated by K.S. Bershowski in 1926, and also emphasized by Lynn Vollen in Symbiosis and the Origin of Species. Vollen produced in 1927 that bacteria might represent the fundamental cause of the origin of species, and that the creation of a species may occur via endosymbiosis. Now, Lynn Margulis, who is also attached to the Gaia theory, claimed that microorganisms are one of the major evolutionary forces in the origin of species. Endosymbiosis of bacteria are responsible for the creation of complex forms of life. So Margulis' theory of symbiogenesis comes down to emphasize that bacteria and other microorganisms actively participate in shaping the earth and help create conditions suitable for life. Eukaryotes require oxygen, and this would only be available after cyanobacteria produced enough. She also argued that these microorganisms still maintain current conditions and that they constitute a major component in the Earth's biomass. She showed that free-living bacteria and other microorganisms tend to merge at larger life forms. In the now generally accepted endosymbiotic hypothesis, Margulis demonstrated that current plant cells result from the merging of separate ancestors, the chloroplasts from bacteria. We look at the origin of some algal and plant cells, the fusion of thermoplasma, spirocheta, alpha, etc., etc., etc. All these different organisms coming together to work together to form and sustain life. She claimed that most of the DNA found in the cytoplasm of animal, plant, fungal, and protist cells would originate as genes of bacteria. Bacteria that became organelles rather than from genetic drift or mutation. With this in mind, Margulis has argued that bacteria have the ability to exchange genes very easily and quickly, even between different species, by conjugation and creating plasmids. For these reasons, the genetic material of bacteria is much more versatile than eukaryotic ones. She argued that versatility is the process which enabled life to evolve so quickly, as bacteria were able to adapt to initial conditions of the environment, and the new ones changed by other bacteria. In the past, public examinations, questions have been posed on this material. This one asks, who proposed that organic molecules could have originated from chemical evolution on early Earth? This was part of the Oparin-Haldane hypothesis, the answer being C. Another question is posed. Who proposed the serial endosymbiosis theory, or symbiogenesis? The correct answer to that is indeed A, Lynn Margulis. This question asks, which scientist demonstrated that a mixture of gas and water could be heated, cooled, and subjected to electrical discharge to generate simple organic molecules? This was the work of Miller and Urey. This question, the constructive response question, asks you to state one way that the theory of intelligent design and the theory of panspermia are similar, and one way in which they are different. The accepted answer for this is as follows. Similarities: both believe that life did not start spontaneously on Earth. Outside forces begin starting things. Differences: intelligent design says all life is created at the same time by a supreme being. Panspermia assumes that life evolved according to the principles of chemical evolution, and panspermia results from life from elsewhere. Thank you for your attention. I hope you found this learning clip to be helpful.